Chapter 9, we're going to talk about microbial growth. So we're focusing on with bacteria. How do bacteria grow? How do they increase in number? They do not go through mitosis, which is what most of you are probably familiar with seeing cells go through. They go through a process called binary fission. They keep it short, sweet, and simple. Generation time is also known as the doubling time. This is the amount of time that it takes for a population to double in number during the log phase. And we'll see this graphed out in just a moment. How long does it take excuse me, for bacteria to carry out their growth phase? How long does it take them to double in number? What is the generation time? That's going to vary quite a bit depending on the species. Some species you might start with a certain number, and for that number to double might take 2 weeks. Some of them, like E. coli, under optimum conditions, it's 18 minutes. So there's a huge range in how long this will take. So this is just showing in this case, it's a Salmonella bacterium, and that over here on the side, you see the picture of having 2 cells together. You're going to have the cell receives a message to carry out DNA replication. That's to duplicate, to make a copy of the DNA. That has to be done before cell division can occur because each new cell needs its own DNA copies. So what happens is the DNA replication will occur. The cell starts to elongate a little bit. Notice here it's circular, but here it's starting to kind of spread out some. The DNA is finishing the replication. Remember, we're talking about bacteria, so the DNA, as you can see here, is circular. The DNA is going to move to opposite ends of the cell. Meanwhile, you've got this pinching in until you have 2 separate cells. So to go from 1 to 2, this whole process, you have a brand new cell. It's going to start over here again. Like I say, it could be 18 minutes if you're talking about E. coli. One thing to keep in mind is, obviously, in microbiology, we're looking at very small things, so you're not going to necessarily see an individual cell. Oh, I've got 2 cells. Oh, look at this. Now I have 4. So we're talking usually about very large numbers, and very quickly they can grow. And so just kind of keep that in mind. In terms of the stages of growth or the phases, there's 4 of them. What we refer to as a bacterial growth curve. You can actually graph these out. You start off a brand new cell, and it will be in the what's known as the lag phase. In this particular phase, there's no increase in the number of the cells because they're adapting the new cells to their new environment. After they have adapted to that environment, then you get this large increase in the number of cells. They're increasing at a constant rate. This is known as the log phase. It's also, some people refer to as the exponential phase because the cells are growing exponentially. Then you're going to reach a point where the number of cells going through cell division is equal to the number of cells dying. This is known as the stationary phase. And then the last phase is known as the death or the decline phase. This is where the number of cells dying is greater than the number of new cells. So if you graph this out, this is typically what you will see. As you start off first, this little low flat line here as it's adjusting to the environment is the lag phase. Here's the log phase where you have that constant rate. During this phase is where you can calculate what the doubling time or the generation time is. The stationary phase is where it plateaus out. And then it starts to come down here. That is the decline or death phase. A chemostat is a piece of equipment. It's used a lot in like industrial microbiology, where they're huge typically, and so you make your media in this big container, this usually like a big vat. So you have all your nutrients here. You sterilize it in here. You inoculate in here. And so what happens is in an industrial setting, okay, in a normal lab setting, you're going to grow your bacteria. And after a certain point, when it reaches that stationary phase, and then the decline phase after that, you're running out of nutrients in terms of the bacteria. And so when you have more cells dying because there's not enough nutrients to keep them alive, to keep them going through their cell division, if you are in an industrial setting where you're wanting the bacteria to produce a particular product, they're going to stop producing that product because they've stopped growing. And so one way around that is to grow them in a chemostat. Basically, what you are doing is you are forcing the cells to stay in that log phase, that exponential phase where you have that constant rate of growth. And how are you able to do that? Is you are able to aseptically, which is sterilely, add in more nutrients, and you remove the product. You remove the waste products that are shutting things down. And so in this type of a situation, because you're constantly adding new nutrients, you're never running out. So those cells just continue to grow and reproduce, and they're making the products that you're interested in, and you keep pulling those. The effluent is what you're pulling out. You're pulling out some of the media that may have some of the dead cells in it. It's going to have, sometimes you're making products that actually become toxic towards the cells where you're removing it, so that never happens. You're removing, remember we talked about feedback inhibition, where you're making a product that then turns off the process. If you remove the product, it's never going to turn off the pathway. So therefore, the cells are continuously making it. So this is, like I said, it's referred to as a chemostat. So great, the bacteria are growing. How do you measure bacterial growth? If you can't see an individual cell, how do you measure the growth of them? Well, you can do what's known as a direct cell count. You can do what's known as a serial dilution. This involves a couple different processes. You can either do a pour plate or spread plate. You might do membrane filtration. You might do an estimate, which is called the most probable number, or MPN procedure, and you might do an indirect cell count using a spectrophotometer where you measure turbidity. When bacteria grow in a liquid broth, they will turn it cloudy when they are growing. If it stays clear, there's no growth in there. That's kind of a little FYI. If you have, say, apple juice in your fridge and it turns really cloudy, I would not drink it because that means there's stuff growing in it. Just FYI. So we go back and we look at direct cell count. You can count the number of cells in liquid culture. How do you do this? You can do a direct microscopic cell count. You have these special slides. They're calibrated. There's like a graph on it, and you put a known volume on one of these special slides. It's very fast. It's very easy to do. It does not distinguish between living and dead cells. You're just doing a total count. Excuse me. You can use fluorescent dyes, which will then distinguish between living and dead cells. Typically, the dyes that you use, the living cells are going to stain green, and the dead cells will stain red. You can use what's known as a Coulter counter. This measures changes in electrical resistance in a solution. Once again, it will not distinguish between living and dead cells. And if it's a very concentrated solution, it's not going to work well. It's not going to be very accurate. It needs to be a diluted type sample. So here's an example of the specialized slides, like I said, that has this grid on it, and you put the known volume in here, and then within each of these grids you count the number of cells, and you can calculate back with a known volume how many you have, and then calculate what the overall concentration is. So once you have one of these slides, it's fairly easy to do. They do this oftentimes. It's similar to counting blood cells in a blood sample. I will say I have one of these slides that was actually given to me when I bought my microscope. It was part of the set. I have since found out that an individual one of these cells is several $100s. So there is an initial startup fee of buying the slide, but once you have a slide, you can reuse this over and over and over. And this is showing the fluorescent staining here, where you can distinguish between the living and dead cells. Once again, the living ones are green, the dead ones are red. So you would put a known volume and you would sit here and you would count to determine in your sample how many cells are there. You might want to do total cells versus living cells, however you wish to do it. This is a Coulter counter. Like I said, this does not work as well with a very concentrated solution, but you're measuring the changes of cells passed through here. Probably a more common way of counting cells is a direct cell count, where you're counting the cells that grow in a petri dish. Obviously, you're only counting the living or viable cells at this point. Now, when you do a direct count using petri dishes, it's very important that you remember you only count between 30 and 300 colonies. Colonies are the individual little spots where the bacteria are growing. The idea is what you see is actually millions of bacterial cells there with the unaided eye, but that it originated from one single cell. So that's how you revert back to it. Now, like I say, you will only count between 30 and 300 colonies. If there's less than 30, sometimes we record this as TFTC, for too few to count. If there's more than 300, we count it as TNTC, as too numerous to count. Some people, instead of recording it as either of those, they just ignore it. You only count between the 30 and 300. Now, usually you have also, with your sample, had to do a serial dilution, so that when you plate your sample on the petri dish, it falls between that 30 and 300. And just so you know, sometimes that's just kind of guesswork to try to get it to fall in there. So you're going to dilute and test within ranges. Once you have diluted your sample, how do you put it on the plate? Typically, you do either what's known as a pour plate or a spread plate. So you will have done your serial dilution where you have your original sample. This is showing a serial tenfold dilution. And so we will talk about this in lab, but just so you know, you're taking a sample, in this case 1 milliliter, out of the original sample. This large jar has your original sample. You are putting that into a test tube with 9 milliliters. So you're putting 1 ml into 9. For a total of 10. This is diluted 1 to 10. How did we get that dilution? The second number here, the 10, that's your total volume. Okay. The 1, the first number, is how much did you put in there? So you put 1 ml in a total of 10. Now, what you're going to do is serial. This means you do a series of these dilutions. You're going to take 1 ml out of this first test tube. You are going to put it in this second test tube that also has 9 ml in it. So as compared to the first tube, you once again did a 1 to 10 dilution, but you're comparing everything back to this original tube. And so now you would, if this was a 1 to 10 dilution, and the first tube is a 1 to 10 dilution, you just multiply them. 1 times 1 is 1. 10 times 10 is 100. And now you're going to do another 1 to 10 dilution. 1 ml into 9 mils. 1 times 1 times 1 is 1. 10 times 10 times 10 is 1,000. And you keep doing this out until, as you see, this last tube here is 1 to 100,000. This will be explained, don't panic, it will be explained in more detail, and I'll work through it a little bit slower with you in lab. Now, what happens here is you are taking a sample out of each of the test tubes and plating it. And that's where you either do a pour plate or a spread plate. In this case, it looks like they took out 1/10 of a mil. So this is complete lawn. You can't see individual colonies. This is so many of them on there. So you're going to say, this is too numerous to count. This is too numerous to count. 389, that's over 300. You're not going to count that one. 50, well, that's between 30 and 300. 2, that's too few to count. So the sample you're going to use to calculate your number as to what the concentration is with this plate right here from the 4th test tube. So you've done your dilution. If you do a pour plate, what you do is you have taken that sample. You will mix it. Usually typically what we do is we'll have in test tubes in a water bath that is kept, typically for us, we keep it around 50 to 60 degrees Celsius. Agar, when you make it and you sterilize it, it comes out of the autoclave and it will be liquid. When it reaches about 45 degrees C, then it will start to solidify. So that's why you need to keep your water bath above 45 to keep it melted. You're going to take your sample and you'll add your bacterial sample to your test tube that has your agar in it. You have to be careful not to add it if the tube is really hot. And this sometimes gets a little tricky because you want it to cool off so you don't kill the bacterial sample, but you don't want it too cool where it starts to solidify too soon. Because you're going to add your sample, then you're just going to dump everything up from the test tube. Just pour the whole thing into the bottom part of your Petri dish. You kind of swirl it around so it completely covers the bottom, and you let it solidify. Word of advice, just let it sit to let it solidify. Do not be in a rush. It is very depressing when you put, as soon as you've poured this in here and swirl it, you're going to have the lid on the Petri dish. And it's just very depressing when you get a little antsy and impatient and you go and you flip that plate upside down, because we always incubate them upside down. And then you watch the whole thing just plop onto the lid because it wasn't fully solidified yet. So just make sure it's solidified. You incubate it and colonies are going to grow. Now, one thing about a pour plate is that because you mix the sample in here and you mixed it well before you poured it, that means that you can have bacteria growing not only on the surface but within the media as well. Sometimes you might want that. For a spread plate, what you're going to do is you have your sample. Now, usually you have your plates pre-made. So you would already have agar in here. You take your sample from your test tubes where you did your dilutions. You put your sample on the plate. Now, this is showing with a glass rod. We call this a hockey stick. You would sterilize the end of this, and then you just spread the sample around the surface. That's one way of doing it. Another way that we've kind of changed, we used to use these hockey sticks a lot, but you have to sterilize them. You take the glass rod, you stick the end portion of this in alcohol and then flame it. And we were starting to have a problem where students were flaming it too long and basically they were all breaking. And a newer, easier way of doing this is you take some small little glass beads. They're in a test tube, so they're nice and sterile, and you just pour the beads on the plate, and then you swirl the plate and it covers the plate. So you end up with the same result. On a spread plate, and oh, by the way, the beads, in case you're wondering what happens to them, then you dump them in a beaker, and we actually soak them in alcohol, sterilize them, and then reuse them. Wash them and then reuse them. A spread plate, one thing that's different about it than a pour plate is that the growth is only going to be on the surface versus the pour plate where the growth is within the entire medium. Another method for monitoring or determining the growth or concentration of bacteria in a sample, and this is something that can be used oftentimes for water samples. Well, they use it for some water samples. The one drawback of the most probable number is that it involves a lot of tubes, so it's going to take up a lot of resources. So sometimes it may be hard to try to sample numbers are low. How much do we have here? So I say it'll be used in water, used in food analysis sometimes too. What you end up doing is you have a total of 15 test tubes. You have 3 different concentrations. So for each concentration, you will have 5 tubes. And what you're going to do is you're going to inoculate each of those 15 tubes that are at 3 different concentrations. You will inoculate them with your water sample or your food sample. And then you will end up with a number. And from this number, you go to a chart and you compare the number on that chart. And you determine an estimate for the number of bacteria per 100 mils, or CFU. CFU is colony forming units per milliliter. I tend to record things as the number of bacteria per mil. One of the other instructors here tends to use the CFU per mil. It's just a personal preference. So with this most probable number, you have that sample. I say you have 3 different concentrations. You use 10 mils, 1 mil, and 0.1 mil of sample. You add that per tube. So you're putting 10 mils in this tube, 1 mil in each of these tubes, 0.1 mil in each of these tubes. These are lactose tubes, and you're looking for fermentation. There is a pH indicator. You're looking to see if acid is being formed. Because there's a pH indicator in it, they start off at neutral and they're red. The phenol red, when it turns acidic, will turn yellow. So you're simply looking for a yellow change. Also, there's an inverted tube in here to collect gas. So you're looking to see if there's gas production as well. And so you would incubate these. Like I say, it is a lot of tubes, or it can very easily become a lot of tubes. After 24 hours, you look at it and you record how many positives you have. So in this first set of 5, you have all of them turn yellow with gas production, so there's 5 positives there. In the second group, you only have 2 that were positive. And in the third group, you have 0 that are positive. So you would record this as 5, 2, 0. And then you would look up that number on your table to determine, and it'll tell you what the estimate of bacteria are for that. There's several different ways of measuring indirectly what the cell count is. You can use a spectrophotometer, which, as I said, is going to measure turbidity or the cloudiness in broth. You can determine what the dry weight of the sample is. You might measure for production of certain products or the disappearance of certain compounds. You might measure the amount of oxygen that's being consumed. So these would all be indirect ways. To use the spectrophotometer, it takes a little bit of work to start off with. What the spectrophotometer does is it will measure the turbidity, and then you relate this to the cell number. So the first thing you're going to have to do with your sample is you're going to have to take it and use the spectrophotometer, get your readings, but at the same time, you're also going to, from those same samples, have to do a direct plate count so that you can correlate. You only have to do this once, though, so that you can correlate. If I get a reading of whatever, 7.2, that correlates to X number of cells. So once you've done this correlation at the beginning, from then on, all you have to do is just read your sample through a spectrophotometer for short. We often just call it the spec. So once you've got that correlation done, then it becomes very quick and easy. If you have a lot of bacteria in the sample, the greater the turbidity, and it means the less light's going to pass through that sample. So the way it works over here on the left is a picture looking down on a spectrophotometer. This is where you're going to put your sample in. You use these special little, they're called cuvettes, look like little square test tubes almost. So you put your sample in that, you close the lid down, and it's going to give you a reading over here on the screen. So the way this spectrophotometer works is that there's a light source in here that is passing light through the sample. So here's your light coming through in this test tube, right there's your sample. If it's clear, which means there's nothing growing in it, all the light's going to pass through. If down here on the bottom now you have another sample here, and look how cloudy it is. Not as much light is going to be able to pass through. A lot of it's going to be reflected off. And so this number is going to be different than the top one. And that's what you're doing, is you're recording those numbers. And because the first time through, like I said, you're going to do direct plate counts, you can correlate it to the number. Once you've done that correlation, this can, if you have a lot of samples, this can be very easy, because you literally take your sample, once you've done the correlation, you put your sample in the cuvette, you stick it in here, close the lid, boom, hit go. There's the reading, open it up, take it out, put your next sample in, close the lid, hit go. It is literally that fast. So you can run through a lot of samples very quickly. Biofilms. This is becoming more of a problem. What a biofilm is, it's a community of different species, different types of microorganisms that literally are growing together as a community, each one helping the other ones out. It's a very highly organized thing. Different environmental conditions will determine what the structure of that biofilm is. Usually what happens is you start off with one organism, and it's growing typically on pipes or tubing. From a medical standpoint, it's a problem with any type of tubing, because the tubing, if it's a catheter or a breathing tube or anything like that, usually it's warm, it's moist, and so bacteria are often thinking, oh, this is a really nice environment here. It's nice and warm, and you're providing nutrients, and I think I'm just going to kind of settle here. And so once one is there, it becomes established there, and then more will come and grow. Other species may come and grow, and so they form, or they secrete, this extracellular matrix that helps them to stick. And other microorganisms are going to come and get embedded in that. They actually kind of have little water channels forming through there. This matrix ends up protecting all of the organisms involved. It's much easier to control by whether it's antibiotics or some type of antimicrobial chemicals. It's easier to control just one or two bacteria, say, on a tubing versus a biofilm, this whole community. That makes it much, much harder to penetrate, have the chemicals penetrate through this matrix to kill all of them. So from a medical standpoint, any type of tubing, like I say, a catheter, a breathing tube, anything like that, you want to leave it in the patient for as short as time as possible. So I say first you have the first bacteria come in, they're going to attach, and then you have additional cells attaching, they're increasing the growth of the original cells, you're producing this matrix, forming these water channels. You continue growing, adding more species, you're sloughing off some old cells that may go establish a little bit further down the tube or the pipe. And like I say, it is very hard to treat and destroy it once it is established. Quorum sensing refers to there appears to be a communication between the cells and the biofilm. It's also going to make it harder to treat. So this is showing down here where you have initially just a few cells there. That's easy to treat. Well, now they're really stuck and growing, and now you've got more growing here, and now you've got your whole little community set up. Obviously, this whole huge setup on the end, on the right, is much harder to treat than if you can get it in the very beginning. So don't let it get established at all. Oxygen requirements for growth. There's a range here, depends on the particular species you are talking about. Obligate aerobes, also known as strict aerobes, they have to have oxygen. They cannot grow without it. If oxygen's not there, they're going to die. Now, on the other end of the spectrum, you have obligate anaerobes or strict anaerobe. Anaerobic is without oxygen, so these are bacteria that they cannot grow in the presence of oxygen. Oxygen is going to kill them. And then we have some that fall in between those two extremes. Facultative anaerobes, they're going to grow best if oxygen is present. But you know what? If oxygen is not there, they're still going to grow. Aerotolerant anaerobes, they don't care. They're going to grow no matter what. You know, take it or leave it, I'm growing. Microaerophiles, these, they need oxygen to grow, but they need it at a lower concentration than what the atmospheric levels are. So it's kind of like, ooh, in the atmosphere, it's a little too strong for us, but we do need it. You can tell what the oxygen requirements are by looking at how an organism grows in a broth. And we will do this in lab oftentimes. The trick here is I always have to remind students when we test this is when you go to look at the tube, don't mix it up. Don't stir it because that kind of messes up everything. An obligate aerobe, because it needs the oxygen, it's going to be growing up on the surface. An obligate anaerobe is going to be down at the bottom, brothers, because there is a gradient or a difference in the concentration in the tube. Facultative anaerobes, remember those, they prefer oxygen, they can grow without it, but they prefer it, so you're going to have growth throughout, but it's going to be mostly at the top. Here, aerotolerant ones, they don't care, they'll grow anywhere. Microaerophiles, they're going to be near the top but not right on the surface, because remember, they're the ones that atmospheric oxygen concentration is a little too high for them, but they need the oxygen, so they're not on the top like the obligate aerobes. So once again, these are all observations that you need to be aware of and notice. You know, so much of microbiology is the observations that you see. And make note of it. Everything adds to the puzzle of figuring out what do you have. Now, if you're talking about organisms that do not like oxygen, so your anaerobes, how do you grow these oftentimes in the lab? There are different ways that you can grow these. You can get special jars to grow them in, where you basically, once you put, in this case, petri dishes in here, then you seal it and then you draw off the oxygen. Sometimes you put in something in there, a type of chemical tube that you break that will absorb the oxygen. In our lab, we tend to keep it short and sweet, and I'll just say we're trying to keep your lab expenses down, so it may be old-fashioned, but hey, it works. What we actually do is we get a large pickle jar, and we put the petri dishes in the pickle jar, and then we get a small candle, and we light the candle, and then we seal the jar. So the candle burns once the oxygen is out, because fire needs oxygen to burn. So once the candle goes out, once you've sealed the jar and the candle's burning inside of it, when the candle goes out, that means there's no more oxygen in there. And then we just put the whole big pickle jar in the incubator. It's not very fancy looking like this, but it works. So that's what we do. Over here on the right side is what is known as an anaerobic box or anaerobic chamber. This is for working with your cultures. There's usually, you can barely see over here on the right side, a supply container that's attached to it. So you would put your supplies in here. I had a friend of mine that used to work with anaerobes, and so she worked with one of these chambers. Once I had to help her with some stuff. Hers was hooked up to nitrogen gas. So she would put her supplies in this side chamber, close the door, flood it with nitrogen gas. So the oxygen leaves, it's filled with nitrogen gas. And then she would come in, put her hands in these gloves. To me, I always found them awkward to work with, but she was used to it. You reach over here, you open a side door between the storage chamber and the actual chamber. You bring your supplies in. This inner chamber is always filled with nitrogen. You do your work, When you're done, any supplies or waste things you put in the side chamber, close the door, take your hands out of the gloves, come back around here, open the outer door, remove your supplies. It takes a little bit of time getting used to it. The effects of pH on microbial growth, the optimum pH, that's where the best growth is going to occur. What pH? Neutrophiles, most bacteria are neutrophiles. They prefer pH around pH 7, maybe a little bit 7.5, usually no more than 8. Acidophiles, they prefer pH where it's less than 5.5. Alkalophiles prefer pH 8 to 10.5. Now you can use pH to kind of help optimize your growth a little bit. What I mean by that is something that's fairly common in a microbiology lab. If you're working with bacteria, usually you're going to be working, as this says, around pH 7. If you're working with fungi prefer a more acidic pH. So if you drop the pH of your media, a lot of your fungal media are 5.5 to 6. Bacteria don't like it that acidic. And so just by changing the pH, if it's a lower pH, you actually inhibit or prevent the bacteria from growing and encourage just your fungi to grow. Fungi don't usually care for pHs at neutral or any higher than that. So when you're wanting to work with bacteria, yes, keep the pH 7, 7.5, and that's going to decrease the amount of fungi. It doesn't mean they're not going to grow in there, but you're going to decrease the numbers. So you can use that to your advantage. Adjust that pH for what you're working with. So this just shows with pH, there's a range of pH of where things are going to grow. So like most of your bacteria are under the screen for the neutrophiles. So like on this, the range they prefer, they can grow in like 6 to about 8.5, but they prefer it up here around 7, 7.5, where the peak is. What's the effect of temperature on microbial growth? Saprophytes will grow from 0 to 15 degrees C. Psychotrophs from 4 to 25 degrees C. If you're not familiar with the Celsius temperature range, 0 is freezing, 4 degrees C is typical refrigerator, about 40 degrees Fahrenheit. Mesophiles, which most of the bacteria we work with are going to be in this middle group, 20 to 45 degrees C. Room temperature, of course, it depends on the room you're in. Room temperature is usually like 21 to 23 degrees C. 37 degrees C is 98.6, our internal body temperature. Water boils at 100 degrees C. So that kind of gives you a little comparison there. So most of the bacteria we work with, certainly if you're thinking medically, is mesophiles. Now there are thermophiles which prefer 50 to 80, and then hypothermophiles 80 to 100 degrees C. That you might find some of these like in your hot springs and Yellowstone up in Wyoming or places like that. So once again, you see these ranges and then the optimum temperature. Some other environmental factors that will affect growth. You have some organisms that are classified as halophiles. That means most organisms don't like a high salt concentration, but these do. They actually require a high salt concentration for growth. Halotolerant means it can grow in a high salt concentration, but it doesn't require it. Barophiles, they require high atmospheric pressure in order to grow. If somebody is collecting samples, say from deep subsurface in the ocean, the pressure is very high there, and so you're going to have to be aware of that. If you want to, that's one reason why it's very hard to bring those culture samples up to the surface and work with them in the lab, is because you would have to maintain them at that high pressure. Phototrophs require sunlight. So most bacteria do require a high amount of moisture. If it's too dry, they're not going to be able to survive as well. When we're growing the cultures, we make the media, the whatever, whether it's a broth or a solid media that we put in a tube, test tube, or on a petri dish. And yes, we're going to come up with names for them. First off, we talk about whether something is a defined medium or whether it's a complex medium. The difference between those two is a defined medium, you know exactly every single chemical and the amount of it that is in that substance. You know that there's X amount of glucose, there's X amount of iron, whatever is in it, you know exactly what that chemical composition is, so what the chemicals are and how much. A complex medium, you don't know what the exact chemical composition is. We have some medium that's known where we'll say it's got yeast extract or beef extract. What is beef extract? You took some beef product and you ground it up and you know it's got some protein in it, but exactly what is it? Who knows? So you don't know the exact chemical composition in a complex medium. An enriched medium is where you have a medium, but you had to add or it contains some essential nutrients that the organism must have. So you enriched it with that. Something that we use in our labs, in most teaching labs, go through a process where we teach you how to use this and how to distinguish things, and that is using selective media and differential media. Selective media is media where some organisms are inhibited while others can grow. Differential media is where you may have different types of bacteria growing, but they're going to change color depending on the media and depending on which bacteria it is. Now, you can have a media that is both selective and differential. There is a type of media in the lab that we abbreviated, we call it EMB. It stands for eosin methylene blue. It has certain dyes in it that will inhibit gram-positives. So gram-positives will not grow. So only gram-negatives will grow on it. That makes it selective. Now there are certain dyes that are added to this so that when E. coli grows, when most cultures grow on it, they kind of look it's a dark media, so they kind of look a darkish color. It's kind of a real dark, almost reddish maroon color, and so that's kind of what some of the other bacteria will look like. But E. coli, when it grows on an EMB medium, E. coli is gram-negative, so that's why it will grow on it. It turns this bright metallic green. There is no doubt about it when you see it. Like, whoa. Well, I can tell just by looking at the color of that one is E. coli. So that's an example of a selective and a differential media, both. In this picture here, this is another one that we work with in the lab that's called a McConkey agar plate. We often just call it a MAC. The McConkey is also selective and differential. It is selective from the standpoint it has certain dyes in the media that prevent gram-positives from growing. So therefore, only your gram-negatives will grow on it. It is differential because if a particular bacteria, and it has to be gram-negative because that's the only thing that grows on it, if it is able to ferment lactose, it will turn this bright fuchsia or bright pink color. So on this particular plate, they did kind of this V shape here with E. coli, which is able to ferment lactose, so it turned it that pink color. Now over here on the top part, you have another gram-negative bacteria, Serratia marcescens. However, that does not ferment lactose. So it grows on the plate, but it stays kind of a cream type, beige type color. It's definitely not this bright fuchsia. So McConkey auger is selective, and that only your gram-negatives will grow. It's differential because you can just look at this plate and very easily distinguish, okay, yes, the bottom half has a lactose fermenter, the top part does not. You can tell that by looking at it. So this just gives you an idea of how we can measure the growth, some of the characteristics of growth in terms of pH, temperature, and things like that. Once again, all helping us to classify and identify the particular organism.